And obviously I'd like to thank the organisers for inviting me to give this talk, and particularly Steve Harridge, although he's not to be blamed if I don't give a good job. So what is gene doping? So this is currently the World Anti-Doping Agency definition of gene doping, which is the transfer of polymers of nucleic acid or nucleic acid analogues, or the use of normal or genetically modified cells. I'm going to stick to the first category simply because I don't think there's enough time to cover the genetically modified cells, but I'm happy to answer questions on that if anybody has any. So what are the concerns with gene doping? WADA has had gene doping on its prohibited list since 2001, and it arises from the concerns that people would be able to gene dope based on the results of studies in laboratory rodents. An awful lot of those studies are in transgenic rodents, i.e. they're a germline manipulation that is passed on from generation to generation, and so the animals grow up with that genetic modification. And that's very different from what you might predict to happen if you were to treat a juvenile or an adult in terms of gene doping. One of the big concerns with gene doping is that most detection methods rely on detecting the difference between a drug and the endogenous product. And that, for instance, we can talk about various forms of recombinant erythropoietin as an example of that. In gene doping, in theory at least, the products are endogenous. They're being produced within the body. They're not being produced from some cell line, maybe of a different species. So that's a concern. And the other concern is that if you gene dope, you could potentially get a nice steady state dynamic which avoids the peaks and troughs associated with exogenous administration of potential doping substances, and that will mean that your signal to noise will be lower and it will make detection more difficult. And then finally, if you're genetically modifying, for instance, muscle, can you detect it using a urine or a blood test, because you're unlikely to be able to go and take biopsies out of elite athletes? So with those thoughts in mind, those are the concerns. What's the actual effect? So if we're going to look at how do we get genetic material into tissues, we need to just consider the vectors that will carry that genetic material in. And the first choice of vectors are the viral vectors. I've listed just a few here, and that's particularly because those are ones that I know have been well tested in muscle. They're replication deficient. They've had all their viral proteins, or most of their viral proteins removed, genes removed, and the genes are provided either in packaging cell lines or in trans with plasmid transfection. And the following just shows that. So you take packaging plasmids, and the packaging plasmids contain the viral genes that are necessary to generate a virus, but what actually goes into the virus is defined by the vector plasmid, which has the packaging signals together with your genetic material of interest. Those are transfected into a cell line or are in a stable cell line, and they produce recombinant virus that can infect once but cannot replicate. And these systems have been developed over the last 20 years, so we have 30 years probably, incredibly safe systems now where the chance of recombination with an endogenous virus are vanishingly small. Just to add to the list, we can also consider non-viral gene delivery. That's based on plasmid DNA, that covalently closed circle DNA that is present in plasmids that can be amplified massively in individual bacteria and can be relatively easily isolated. Compared to a viral vector, these are incredibly inefficient vectors. You can compound them with lipids, with proteins to try and target them. You can try and make artificial viruses. Any attempt we have to make artificial viruses are pathetic compared to what the viruses themselves can do. The one thing that does improve delivery substantially, particularly to structures such as muscle, is if you combine them with some sort of energy process, be it a high pressure delivery, be it electroporation or ultrasound. Projectiles are good for skin gene transfer, but not much use for the muscle, I think. So there's a list of potential gene doping candidates, and this is not an exhaustive list, this is just meant to be illustrative, but you can have genes that will increase muscle mass, And obviously things like growth hormone, IGF-1, and the blocking of myostatin would be examples of that. You can have genes that will improve the cardiovascular system or cause the induction of greater numbers of red blood cells, such as erythropoietin and VEGF. You can modify the skeletal muscle phenotype by either stimulating the slow aerobic phenotype for increased endurance or activating the fast muscles for that fast phenotype associated with the power sports. And then finally, and I'm not going to touch on this because the data is very limited in this area, is you can potentially raise the pain threshold and therefore enable people to undertake greater degrees of effort. This really is where the problem originally came from. The demonstration that if you made a transgenic mouse overexpressing IGF-1, that it showed marked muscle hypertrophy, and that was early as the mid-1990s. Perhaps the most critical paper then was showing that you could not only do it in a transgenic, but you could take a viral vector, deliver that to mice, and show that not only did that increase muscle mass in young mice, but importantly, it countered the age-related decline in muscle function in older mice. And then finally, the other paper which really sort of stirred up this area was showing that if you gave IGF-1 to rats as a gene transfer, and then you made them train, and that's what this rather gross picture of a rat climbing up bars with a whole series of test tubes attached to it as a weight. I think we'd be pushed to get that through the Home Office in the UK now. That showed that you could have an additive effect. So the IGF-1 boosted the training effect. But in all these cases, the IGF-1 had to be delivered to the muscle, had to be present in the muscle. I think we're all probably fairly familiar with the myostatin story, but just to summarize that, myostatin is a negative regulator of muscle growth. We have a number of natural and induced mutants where the myostatin gene has been inactivated or is naturally inactivated. So you get the double muscling that occurs in some of the continental breeds of cows, such as the Belgian Blue. But you also have the demonstration of a direct knockout in mice that produces a marked phenotype between littermates, depending on whether they're knockouts or not. Perhaps more interesting in the myostatin story is the variant we see in whippets, where there is a myostatin mutation that, when it's present in 2 copies, gives you the bully whippet, which is a hyper-muscled whippet that really isn't much good as a racer because it's just too chunky and it doesn't perform particularly well. But in the heterozygote state, this myostatin mutation gives more muscle and faster racers. And now we start to see where probably the problem is. We don't want to knock out myostatin altogether, we just want to knock it down, and then we should get an increased performance. And interestingly, in horses, if you take a particular mutation that's present in the intron 1 of the equine myostatin gene, you can actually show, when you look at racing records, that particular variants of that SNP are associated with good performance in different sports. And so that really, again, tells you that the potential for manipulation of the myostatin system is really quite a powerful target. So just to summarize, the myostatin null animals do tend to have metabolic problems. They really are not very good performers, so that's clearly not something we're aiming to achieve. The heterozygotes, as I showed you, have performance advantages and the subtle effects for the performance in horses. There's a lot of ways of reducing or blocking myostatin that are being investigated, because clearly if you could do this, it would be great for people with cancer cachexia, it would be great for people with prolonged bed rest, it might be the solution, at least in part, to sarcopenia in old age, and of course it would be useful potentially in muscular dystrophy and other related genetic disorders. So there's a huge amount of work going on in this area, and if we take at least a gene therapy approach to this, then potentially there is the option of taking that through into athletic doping. Again, I don't want to dwell too long on EPO, but just to make the point that obviously blood doping or using erythropoietin to induce more red blood cells seems to increase athletic performance in a fairly convincing fashion. And that, without doubt, if you wanted just in terms of the ability to do that gene doping is the easiest thing in the world. Whether you can actually then get away with it is a different matter altogether. And then modifying the muscle phenotype is actually a more controversial area. So if we look at transgenic mice that are expressing PPAR delta, they have increased performance. Mice that are overexpressing PGC1 beta have increased endurance at high load. Transgenic mice with PGC1 alpha or PepC-K also have an increased aerobic capacity. But all of those are mice that are genetically modified and grow up with those additional genes, and in many cases those genes are overexpressed at quite substantial levels. What we have is very little evidence that taking the same genes, delivering them at a relatively modest rate to adolescent or adult rodents actually makes a major difference, and that's where the literature is very sparse. Could we detect gene doping under the situations I've just given you? Well, I think yes. I think the basis of doping detection right now for exogenous substances is really a direct detection. And that's becoming an increasing problem because you have to know exactly what you're trying to detect, and we know the history of drugs that have been developed that escape the initial detection. Biotechnology products, such as those associated with gene doping, ought to be readily, easily detectable. For instance, one can use a quantitative RT-PCR, or you can do a nested quantitative RT-PCR. You can get incredibly sensitive in terms of detection, but again, you need to know what you're looking for, and that's perhaps the biggest drawback in combating gene doping. For those proteins that are expressed systemically, such as erythropoietin, we know that post-translational differences may enable us to detect those, particularly if they're being expressed from ectopic sites, in other words, not being produced from the kidney where they're normally produced. We can obviously look at biomarkers, and we have the athletic passport, and all of these are probably quite good approaches to catch potential genetic cheats. And I think it's fair to say in this field, there have been more review papers and more papers on detection of gene doping than have actually shown the real potential for human gene doping. It's implied from the transgenic studies, it's not proven in most of the other cases. This is just a classic example of differences in erythropoietin post-translational modification. This is the original paper that pointed this out, that you could have on the left-hand side recombinant EPO produced from various cell lines, and they have a particular pattern, isoelectric focusing pattern. If you look at normal erythropoietin produced in humans or in mice, they have a very different pattern. And then if you do look at the ones post-gene transfer into muscle, again, you have this abnormal pattern. This has been quite controversial in places, but I think with the series of papers that have been published by this group, it's now fairly clear that this is still holds true despite concerns about particular antibodies and about particular cross reactions. So why don't athletes gene dope at the moment? Well, as far as I'm aware, there is no evidence that gene doping happens in athletics right now. And the question is why? Well, the first is the technology. It's very easy to genetically modify a mouse, particularly if you're doing a transgenic experiment. It's relatively easy using viral vectors now to genetically modify mice. It's a bit trickier in rats. And as you start to go up through the size, you come across a number of problems. One is simply producing enough virus. Producing these recombinant viruses is not trivial. It's trivial at the bench scale enough to do a cell culture experiment. It's moderately trivial to do it for a mouse experiment. It's a real issue when you want to do, say, for instance, gene transfer in dogs. And at the moment, we are right on the edge of what's possible in terms of thinking about gene transfer in humans. It's only in the last couple of years that the scale-up in the really very best centres managed to get to the point where it's feasible to consider this sort of approach. The technology not only is a problem for scaling, but there are lots of other things that we need to consider. The efficiency of packaging of the recombinant virus is remarkably poor, so an awful lot of what you deliver is actually a non-functional version of recombinant virus. Again, we're getting better. The last few years have seen some significant improvements in that. Even with all of that, we have to give large doses of viruses in order to get a relatively good gene transfer. So the problem would be at the moment with, say, the AAV technology, which is pretty much state-of-the-art, this isn't something that somebody could do in a backroom lab. This requires major fermenters, it requires major column purification. It's the kind of thing that is within state-sponsored gene doping potential, but certainly not at the level of an individual rogue coach or whatever. And then the other problem is detection of product. It turns out quite easy relatively easy to detect the presence of synthetic genes, for instance, cDNAs, which is what so it's the coding sequence of a gene, not the coding and non-coding sequences, which is what you get in the genomic region. The cDNAs will not be there normally, and therefore their presence is fairly easy to detect. That's particularly true, again, if you can also detect the protein product if that's being secreted. Probably our most worrying area is those things that go into muscle and stay in muscle and that rarely escape from muscle. Despite that, a number of studies have shown fairly convincingly that at least within 4 to 6 weeks of the initial gene transfer, you can pick up sufficient in the plasma in order to be able to detect it. And then finally, whilst we understand quite a lot of regulation of gene expression, packaging that all together in a viral vector is tricky, and then guaranteeing the efficiency of that viral vector delivery to tissues is almost impossible. So we'll get some cases where we get a very good delivery, other cases where we get a relatively poor delivery, and so we will have very different levels of gene expression depending on each individual case. But I think that's going to change relatively soon. There are a number of technologies that have matured in the last 2 to 3 years which enable us to, which would concern us about the potential for gene doping. One is the ability to rather more accurately genetically modify a desired piece of the genome. The second, as I alluded to earlier, is that the manufacturing issues associated with producing sufficient recombinant virus are largely solved, at least in the very best labs, and that within the field I work in, Duchenne muscular dystrophy, where I suspect most of the information that is going to be used potentially for gene doping will come from, in DMD we're due to have systemic clinical trials of viral gene delivery in the next 18 months. Maybe that's not going to be sufficient data to tell us exactly what we need to do in order to have efficient gene delivery, but it'll get us quite a step towards that. So what are the technologies that are emerging that potentially have the greatest concern? Well, one is the TALEN technology, and the other one is the CRISPR-Cas9. And they both basically depend on the ability, using sequence-specific nucleases, to create double-stranded breaks that enhance local DNA repair and allow recombination of oligonucleotides that you can add into that repair process. And it's orders of magnitude more efficient. It's probably in the order of 10 to the 3 more efficient than we've been previously working with, but it's still probably only at the level of about a 1% efficiency. The other thing you can do with the double-stranded breaks, if you don't provide something to direct a degree of homologous recombination, is of course you can have non-homologous end joining, and essentially what that's going to do is to introduce a mutation in your targeted site. And the TALEN technology depends on transcription activator-like proteins, which are individual peptide motifs that recognize single nucleotides. And so if you combine those individual peptide motifs, you can target a specific sequence. Add to that a nuclease, in this case it's the FOC1 nuclease, and then you have something that will sequence specifically cause a double-stranded break. And the technology that you probably heard rather more about in terms of being in the news is, of course, the CRISPR-Cas9 technology. It's a similar sort of concept, except that what you provide is guide RNAs in order to target the specific sequences that are then acted on by the Cas9 nuclease. And in the case of the CRISPR-Cas9, that's now been used in allegedly adult mice, but I happen to think a 6-week-old mouse is a juvenile mouse. And this is just one example from that paper that came out in Science at the very end of last year. There was actually a set of 3 papers, but this was the one that actually showed that you could potentially deliver the CRISPR-Cas9 systemically in at least a juvenile animal. And what we see here is evidence that you've got the specific modification, which is actually getting rid of exon 23, where the mutation resides in the dystrophic MDX mouse. You can show that some dystrophin is produced, and here we have a cardiac muscle. So this is cardiac sections. This is wild-type dystrophin expression, so that you can see these green circles are dystrophin just under the sarcolemma of the myocytes. This is the MDX mouse muscle, and this is the MDX mouse treated with CRISPR-Cas9, showing that you have indeed managed to restore dystrophin. Not at very high efficiency, but it's a start, a proof of principle. So how would we apply this to humans? Well, we can go back to examples of specific human mutations associated with increased athletic performance. And this chap, whose name I won't attempt to pronounce because I almost certainly mangle it, was a high-performance athlete, admittedly a rather large number of years ago, and it was shown that he carried a genetic mutation that meant that he got an overproduction of erythropoietin and therefore had more red blood cells. You could, in theory, induce exactly that mutation or a related mutation using CRISPR-Cas9. Same way, you could create mutations such as I've described in dog and in horse in the myostatin gene to disable that gene. And what's more, those mutations do not include the addition of any exogenous sequence. So as such, they will potentially be undetectable. Okay, what will probably happen due to the efficiency of gene delivery is it will actually find that these individuals are chimeric, but just because you're chimeric doesn't mean you're cheating. So I think it's something we certainly need to watch out for as potential. So, as I've said, it'd be difficult to prove them as doping. You don't have an abnormal secreted product because, for instance, if you did a mutation in the EPO gene, it's still going to be produced from the kidney. It'll still have the same post-translational modifications, so you can't claim it's a doped product. And if you just watch the field of CRISPR-Cas9, it's exponential, the rate of development of that field. Every month we're seeing new and improved vectors. But, as I've said before, if you're going to deliver this, you'll have to deliver it probably with a viral vector, and we know that will require a huge amount of virus. Critically, with a viral vector, in most cases, re-administration is not possible. And that's just based on the studies in mice and in dogs. If you go to the human population, many of humans have already experienced these viruses. They're actually non-pathogenic viruses that cause no problems, but do induce an immune response. And therefore, some people at least will be refractory to treatment. And at the moment, the system that's being used is a DNA version, a DNA encoding for Cas9, so that DNA will be present and the Cas9 will be present, which means you can detect that DNA potentially, and you have the activity of the Cas9 protein bouncing around in your cells, which increases the risk of off-target effects. This is a particular problem if you're thinking of applying it to a whole organism. If you do it in cells in culture, the cells divide, AAV doesn't integrate, and therefore the genome is lost. But in muscle, there's no division within the muscle fibers, and that DNA is retained for very, very long periods. Obviously, there are safety concerns with gene doping. There's potential for innate immune responses to the viral and non-viral vectors, and if you've been exposed in the past and now you have your first administration, that could lead to all kinds of problems, including a massive cytokine storm, which we've actually seen in gene therapy trials. When you're doing things like EPO gene transfer, particularly if you're doing EPO gene transfer to an ectopic site such as muscle, potential for autoimmune responses demonstrated in non-human primates. Polycythemia is always a worry, but of course that can be controlled with regular medical checks. And then, as I'm sure you're all well aware, there's potential oncogenesis from a number of the factors that one might think about introducing by gene doping. So, is it a current risk? Right now, I couldn't tell you whether we're going to be able to get AAV delivered systemically at any sort of efficiency in man. We don't have that information. But as all the other barriers are being solved, I suspect that that one also will be. The focus should obviously be on generic detection approaches, and there's a number of approaches out there which I think will help us to at least combat the initial potential application of gene doping. But it's really important that regulators are on top of the state of the art. And I think, as I mentioned before, genetic manipulation within muscle cells, it doesn't get out of the muscle, is going to be critical. And I expect that over the next few years, I very, very much hope we'll see major progress in muscular dystrophy, and that will certainly increase the risk of gene doping. And with that, I thank you for your attention.